Good morning, Dr. B, back once more. I thought I would add a third video to Chapter 8 on photosynthesis to try to explain the different means of carbon fixation that are entitled C3, C4, and CAM photosynthesis, and try to explain what those three variants are. They all kind of revolve around the enzyme rubisco. And rubisco is an acronym for ribulose bisphosphate carboxylase oxygenase, a large complex enzyme that's considered to be one of the most abundant proteins here on Earth. It is the enzyme that fixes carbon dioxide into a pathway to eventually form an organic molecule. So all consumers require this particular enzyme for their eventual source of energy, because we consume energy that producers produce. Rubisco is thought to have developed in an environment that is anaerobic. And that really is one of the key issues as to one of the problems that it has in trying to fix carbon dioxide in an environment that today is basically aerobic, oxygen being present. The last bullet and the figure of rubisco show it to be a very complex protein, and the different proteins that are involved in the rubisco molecule are synthesized from genes that are contained in both the chloroplast and the nuclear DNA. cDNA, chloroplast DNA, and nDNA is nuclear DNA. I put the slide of the Calvin cycle, or the dark reactions of photosynthesis, back into this video just to show you the location of rubisco at the very top at 12 o'clock of the Calvin cycle. It fixes carbon dioxide, and the first stable molecule that's formed contains 3 carbon atoms and is called phosphoglycerate, or PGA. That is thus used in the designation of a C3 plant, that the first stable molecule formed in the Calvin cycle or dark reactions is an organic molecule with 3 carbon atoms, so C3. Rubisco is responsible for bonding carbon dioxide into this cycle. The issue results when in C3 plants, oxygen levels may become higher than carbon dioxide levels within the leaf of the plant. The conditions of the climate where this might occur is when the climate is really hot and dry in the summer. So think when it's hot and dry, a plant might decide to close its stomates and prevent the movement of gases in and out of the plant. That would prevent the release of oxygen and also prevent the entry of carbon dioxide. In this regard, the plant would have a higher level of CO2 that could build up in the leaf compared to carbon dioxide. And rubisco, unfortunately, cannot distinguish between oxygen and carbon dioxide. They're actually very similar sized molecules. The only difference is carbon dioxide has a carbon atom. Oxygen doesn't. So, if oxygen levels are higher than carbon dioxide levels in a leaf, the enzyme rubisco can actually use oxygen and bond oxygen into the Calvin cycle or the dark reactions. This will only form one molecule of PGA, phosphoglycerate, and it forms a second molecule that's called phosphoglycolate. You're like, oh Lord. It's really not that difficult. Phosphoglycolate has to leave the chloroplast. As a matter of fact, it actually then travels to an organelle called the peroxisome. You've heard of it as an organelle from chapter 4 that detoxifies hydrogen peroxide. The peroxisome will bond two phosphoglycolate molecules together to form PGA, but in the process it will release a molecule of carbon dioxide. That's where the word photorespiration comes from. Once the peroxisome has produced PGA, it is then moved back into the chloroplast, into the stroma of the chloroplast. So, in days that are hot and dry and the plant closes its stomates, in plants that have what is called C3 photosynthesis, PGA is the first stable molecule formed. These C3 plants can undergo photorespiration. Again, hot, dry, environmental conditions, plant closes its stomates, photorespiration is possible. This may reduce that plant's production of sugars, carbohydrates by up to 50%. The explanation for this photorespiration issue is that the enzyme rubisco probably evolved when there was very little oxygen present in the environment. So, because of the similarity between carbon dioxide and oxygen, the active site on the rubisco molecule cannot distinguish between those two molecules. So photorespiration is an issue in C3 plants. The environmental conditions for this photorespiration, hot, dry climates, think deserts or open grasslands, prairie. So photorespiration, that means the release of carbon dioxide, and this release of carbon dioxide is basically being driven by light, photorespiration. So instead of carbon dioxide entering the Calvin cycle or dark reactions, it is oxygen that enters. The reduction in glucose is up to 50%. The reduction in the production of glucose is up to 50%. The environmental conditions when this process of photorespiration might occur, hot, dry, perhaps even water being lacking. You know how droughts are common in the summer months. Stomates may close on a plant that has C3 photosynthesis, and the production of glucose can be severely limited, the production of its energy supply, because the supply of carbon dioxide from the air has been cut off when the plant closes its stomates. So these last slides now talk about, well, what's the solution to photorespiration? The solution is to fix carbon dioxide in a different manner. And we refer to this as a C4 pathway. And what that means is, look at number 2. Well, let's look at number 1. The first acceptor of carbon dioxide is a 3-carbon molecule that is called phosphoenol pyruvate. So there is an enzyme that will bond carbon dioxide to phosphoenol pyruvate. The acronym for that is PEP, PEP. So CO2 bonds to PEP and forms a stable product that is called oxaloacetic acid. Oxaloacetic acid contains 4 carbon atoms. So this solution is called a C4 pathway. Oxaloacetic acid then gives up a carbon dioxide molecule directly to the Calvin cycle, such that rubisco, which is used in a C4 pathway, will only bond carbon dioxide into the Calvin cycle. So it is a very efficient process of only fixing carbon dioxide into the Calvin cycle, occurring in environments that are hot and dry and perhaps even deficient in water, such as a desert or a dry grassland or prairie. In some plants, these are mesophyll cells. These cells here that surround this vascular tissue, these cells here are called bundle sheath cells. So plants that have the C4 pathway often have photosynthesis, the dark reactions or Calvin cycle that occur in these cells that surround the vascular tissue. That's another modification that they have. So plants with the C4 pathway often suffer very little photorespiration. Examples of plants with the C4 pathway include plants that are succulent, like cactus plants, grasses that grow in the hot summer months, corn, which is illustrated on this photo, and pineapple. So the C4 pathway involves carbon dioxide being bonded to a molecule called PEP. That forms oxaloacetic acid. Oxaloacetic acid releases a carbon dioxide directly into the Calvin cycle, where then the enzyme Rubisco will bond it to form PGA, and the Calvin cycle runs at 100% efficiency. There is no photorespiration, very limited photorespiration. Plants that have traditional C4 photosynthesis first fix carbon dioxide in a leaf cell called a mesophyll cell, and then release carbon dioxide into the Calvin cycle in the bundle sheath cell. So there's two different cells involved in plants such as corn. In plants such as pineapple, they fix carbon dioxide at night in a mesophyll cell and then release carbon dioxide during the day when their stomates are closed. So the cell is just one single cell where the process occurs. In both types of plants, the C4 molecule is called oxaloacetic acid. When it releases CO2, the molecule that forms from that is PEP. So you notice in both cases you have two cyclical pathways, a C4 pathway and then the Calvin cycle or the dark reactions. So my last slide here is a comparison. About 90% of all plants have C3 photosynthesis. When carbon dioxide is fixed, the first molecule is called PGA, phosphoglycerate. The enzyme, there's only one, Rubisco. Photorespiration can reduce sugar production by up to 50%. It's primarily only a problem when conditions are droughty, and that primarily occurs during hot, dry weather during the summer months. Plants that live in open grasslands, two of our most important crop plants are corn and sugarcane. They have C4 photosynthesis. Phosphoenol pyruvate attaches to carbon dioxide via the enzyme PEP-ACE and forms oxaloacetic acid. It then releases CO2 into the Calvin cycle, and the enzyme Rubisco is also present and is used in the Calvin cycle. CAM plants are plants like succulents, like cactus plants. They fix carbon dioxide at night and release it into the Calvin cycle during the day. I hope this is a better explanation.